Good evening, one and all. And it's been a pleasure to have you at this another session of Envision with BDG Life Sciences initiative. Uh, I can see that there are many new participants who are joining this session, so it's really good. And I can see many old participants also. So well, that is great. Great to see the consistency of the participants, and I'm thankful to all of you for again taking out your time in attending the sessions. So today's session is a really interesting one, and as we wanted to diversify the topics, hence we have included these topics of artificial intelligence and machine learning. However, we are doing a lot of programs like courses and as well as workshops in artificial intelligence in biology, artificial intelligence in drug discovery. But then this, we want to give you something more. So that's why today we are having Dr. Haider from United States. United Kingdom, and he will be definitely speaking on the brain-computer interfacing. He is an expert in his field, and I will tell you about him later on. First, as you all know that like people who are not new to this, so who have been attending regular sessions. So first of all, I have to start off with the company presentation. However, I will not take much time because most of you have already been known and are being consistent in our program. So I will not take much time of yours in just talking about the BDG Life Sciences, who we are, and why we started this kind of sessions or these type of sessions. So just to give you the idea that why we started Envision with BDG. Now, as you all know that BTG Life Sciences is a 10-year-old company, and we are doing online trainings and workshops since 2012. However, we didn't what I should say, we have been doing a lot of online programs, since a very long time. But then since this COVID-19 pandemic started in February, in March, we have increased the frequency of our online programs. And then previously we used to launch free courses, say like one course in 2 months. But then since this pandemic started, we have conducted more than 70 courses, and in which more than 14,000 people from all over the world have participated. So this is something of which we are proud of, that people, we've helped people in providing free courses. So then after 6 months or 7 months of rigorous efforts, we thought that why not to do something new. So I personally started, and I asked my team members to search people in their connections, and those people who are renowned and who are experts into their fields, so that the idea was to give you more, because this organization is all about it's a group, and we have not you can find it group in the name of the company. So it is for everyone. That was the motive of starting the organization. So that is the reason why we want to give something more. And then for that we have thought that okay, fine, let's find our people in our connections and ask them to deliver online lectures or sessions where they can talk about their expertise. But and you know what, it's very good for all these kind of people that they have turned up, and they are doing the sessions in taking out time for us, so that you guys can have a good learning experience. Now these sessions are available on our YouTube channel also, so you can anytime access it. So let's continue with BTG Life Sciences, who we are and what we are. So basically, we are a guide who help you in solving your academic problems. So this is was the motive of starting the organization, which means to help. So we started this organization in the form of with the motive of helping young students, research scholars, and especially the students who at one point of their time in their career, in their bachelor's and masters, they do not understand, or they stuck at a point where they do not know what to do next. And that is the reason they used to change their field, or do something which might not result, or which might not be a good opportunity for them. So later on, they used to face these job crises, and that is the reason why we started this organization. So the main 5 points on which we are focused on is like how we are helping, showing you the right path, solving your queries, giving you correct training, and then paving your path for a successful career, and giving you launchpad to excel into the field of science. So these are the 5 pillars of BDG Life Sciences, and we have mixed it up, or I should say cemented it, with innovation and creativity, and that is how we have been successfully running this organization since 10 years. So in 2009, we started in the form of a group, and the motive of the group was to help people. Now what this group had free consultation, this group had project reports, this group had training updates, this group had free training, this group had free the what I should say is, we used to provide PDFs, and then most importantly, job updates available in India as well as abroad. So the targeted audience for this group was professionals, students, and research scholars from biotechnology, bioinformatics, and pharma. So this was the motive of starting the group in 2009. Back then, when we started this group, it was around a few people on Google groups, but now we have more than 1 million subscribers from all over the world, and these subscribers are not only students, but they are faculties and company professionals. So this is what we have achieved, and now we have transformed ourselves, means the group has been transformed into what I should say, Facebook, WhatsApp, Telegram. So a number of platforms are available where we are providing, where we are helping the students and the research scholars, and paving their path for the successful career. So these are some of our subscriber platforms. You could see, Facebook, then LinkedIn, YouTube, Twitter, Instagram, and then on Messenger we have WhatsApp, Telegram, Google Duo, and What I Am. And then we have others like Pinterest, Flickr, etc., etc. So you could also see that we have our own website, where we have a dedicated page where you can go on and provide your details and subscribe to our newsletter, and then you can communicate with us. So this is what our subscriber platform looks like. Now comes to the business. Now this, when we are talking about the social work, this is the social work which we have been doing, but then definitely we have to survive, so we are doing the business. Now the business is done in such a way, like from the beginning. In 2010, when we started this company, the name of the company was Biodiscovery Group only. But then in 2016, when we changed it to BDG Life Sciences, then also we have not removed the group. So we wanted to stick to our roots, and the roots was that it was a group and it will be the group. So that means that we are for everyone, we are helping everyone, so this company is for everyone, and that is the motive of the organization. So the first program which we started was the research project program, in which we have launched novel research projects, and then created a business where it was or I should say, it is a win-win situation for both the company as well as the clients. So these novel research projects help you in working, or I should say, getting trained into the latest technology. Plus, you are working on the project, hence you are having the practical application, and then international publication of the project, which is strengthening your profile. So what you are getting training, number one, practical application, number two, and number three, the most important thing is that is international publication. So this was the motive of starting the business of BDG Life Sciences or Biodiscovery Group, to create a win-win situation for both the company and the client. So you pay us the fee for the project, and the fee is not very much, but then yes, it varies from project to project. However, you can just check out the research projects page. Right now we have a project on anti-cancer drugs, so molecular modeling project is there, so you can check out the research projects page of the website, and there you could see the fee details. So however, what I was focusing over here is that creating a win-win situation for both of us, means you are getting training, practical application, as well as international publication, which is strengthening your profile, so that you can have much better job opportunities. Now, as the time has progressed, we are a 10-year-old company. Hence, for example, people who have joined us, say in 2010, they were doing their masters or they were doing PhD. But right now, they have become, some big scientist, or I should say, they are at a very big position into the company, or some of them are running their own company, which means what? That they are still connected with us and they know the quality of our work. Hence, they ask us to, to send people for employment, to recommend people for employment. So this is what we are doing. We are not promising that after you do this program with BTG Life Sciences, 100% job assurance, 100% job guarantee. We have never advertised any of our program in such a way that after doing this particular kind of training or this particular kind of a job, you will get 100% job. No, this is not what we are doing. But what we are doing is that we have people from our connections whom we recommend for PhDs as well as research scholars. So this is how a circle gets completed. Means, for example, today, if you join us, after 10 years, you become some great scientist, or someone good into the field, and someone else joins after 10 years, you ask us for recommendation and we send you that particular person's contacts, and this is how we connect people. So this is what we are doing. So training, practical application, international publication, and then recommendations. And obviously on LinkedIn, we are writing recommendations for you, so that it's a win-win situation for both of us. So our wide array of services includes contract research, workshops, e-workshops, that is online workshops, research projects. I've already told you about that, internships, and then training, webinars, seminar, and conferences. So this is the business part of biodiscovery or BDG Life Sciences. Then you could see over here that this is the slide which tells you about the kind of business which we are getting from around the world. So the most type of business which we are getting is of research projects. Why? Because this is of novel research projects. Everyone wants to do research and wants to get international publication. So to have much better opportunities, whoever, if they want to do a PhD or if they want to do their major project or dissertation project or thesis project, this is the program which they choose. Now, as it is online also, like we used to do it on site as well as online, and we are doing it since 2010 when we started the company. But then as it is online, so people from all over the world can participate. So the project team is diversified, means someone is working from Egypt, someone is working from Australia, someone is working from India, someone is working from the United States. So the team is diversified, and there are different level of people who are working on the same kind of project, and we've made a one team. For example, one person is a PhD scholar, one person is an MSc intern, one person is an MTech, and one person is a bachelor student. So this is the diversity of the kind of team which we have for our research projects, and that is why this project means this research project program is a big success. Then we have workshops. These are the on-site workshops which we used to do in India as well as in other countries. However, since COVID-19 pandemic started, we postponed all our on-site workshops till December 31st. Then e-workshops, online workshops, again, it has opened the window for people to participate in by online mode. So that is why this graph is also up. And then courses, these are the free courses. This has the highest number of participants. However, we are not earning from this because we are doing it for free. Then we have others like contract research and project designing. So these are the this is what exactly the business of BDG Life Sciences. The mission of the company is to catalyze the process of biomedical sciences into therapeutic technologies that benefit human health, most importantly, and eventually fuel the economic growth. So this is the mission of BDG Life Sciences. And as I have already told you, innovation and creativity is the key pillars, or I should say the cement of BDG Life Sciences. Hence, we keep on introducing new programs. Like in 2019, we introduced artificial intelligence and machine learning, artificial intelligence in biology. And then only after 4 months of that program, we launched our program of artificial intelligence in drug discovery. So these are the programs which we are doing. And then we were the first one to to launch and conduct online as well as on-site training on gene and genome editing using CRISPR-Cas9, for which the two women scientists, Dr. Jennifer Doudna and Dr. Charpentier, has got the Nobel Prize for Chemistry this year. So in 2017 May, we were the first one to launch the training on and workshop online as well as on-site of CRISPR-Cas9. So these are the topics on which we are doing our work, like bioinformatics. It has two modules, basics as well as advanced. Then gene and genome editing using CRISPR-Cas9, Cas12a, Cas13a indicases, and then drug discovery technology. In this, we are doing two kinds of, in fact, three programs we are doing. One is normal drug discovery. Then other one is virtual screening and molecular docking of MPRO from SARS coronavirus. And then another workshop which is already which is currently running, and that is drug targets of SARS coronavirus. Then we have molecular simulations and dynamics, predictive toxicology, a number of NGS data analysis. We are doing whole genome, exome, transcriptomics, metagenomics, and then yes, artificial intelligence and machine learning and biology, which includes drug discovery itself. So these are some of the programs. And then we have disease specific research programs like cancer, HIV, diabetes, and tuberculosis research. So this is what our topics include of research projects, workshops, online workshops, and training. Now, this is the most important slide, and then I will quickly run through this presentation and then hand it over to Dr. Haider. So, value proposition. Now, in this, all I need you to focus on these four words: innovation, ideas, service, and value. Now, if we want to read it out as a sentence, it says that innovation is to translate ideas into service that creates value. So as I have told you that innovation is the integral part of BTG Life Sciences. Hence, what we are doing is that we are innovative in our ideas and providing excellent service to which is creating value for our customers. So this is what we are doing since 10 years, and that is the reason why we are still at the top of the bioinformatics service providers in terms of providing trainings and research projects. So what exactly we are doing? This is what we are doing: practical application of technology. Now we all know that we learn by doing the things. Hence, we have included practical application of all our training programs. Like, it's not about just PowerPoint presentation and then that's it. No, it's not like that. We will make you work, and that is the reason you could see over here hands-on training session. So whatever program you are joining, you will be we will make you work. And when you will work, you will encounter problems. Once you will encounter problems, then you will come up with your solutions, and that is the problem solving as well as the learning procedure. Then novel research projects. Novel research projects is the key, because this is what we are giving, and you might not find it anywhere. And then result driven research and everlasting relationship. Everlasting relationship, I have already talked to you about, that people who have got connected with us 10 years before, they are still connected with us, and that's the way we are helping them. So they keep questions and we keep on helping them. The next slide is the achievements, and you could see, I'll just only say that if we would not have been innovating, then at one point of time this graph would have come to saturation and then it would have dropped down. But then it's not, it is going up, which means that we are innovating, and that is the reason why it is keep on going up. So in a nutshell, what I would like to tell you is that we have 10 plus years of experience, and then 60 plus research projects, 400 plus workshops in India, 20 plus workshops in other countries. That include, just to name a few, Mexico, Iran, Saudi, so not Saudi Arabia, Egypt, Kenya, Nigeria, and Sudan. Then comes to Dubai, then Thailand and Malaysia. So these are the countries where we have been doing our regular workshops. Then we have been doing the regular workshops at premier institutes of India like Indian Institute of Technology, Guwahati, NIT, VIT, MMC, and then All India Institute of Medical Sciences, New Delhi, etc. And then happy customers. So happy customers, feedback is an integral part of BTG Life Sciences, because we believe that the communication process is not complete unless you get the feedback. Hence, when we started the organization, in the beginning, we used to have feedback forms. Then we transformed as the technology came, and then we transformed into asking for the feedback at the website. And then now we have video feedback. So on our YouTube channel, you could see that we keep on uploading the videos of our participants, so that you can see that what was their experience when they have done the training with BDG Life Sciences. So I've already told you about that we were the first one to introduce training in drug discovery and online training in drug discovery and designing, and then gene and genome editing. And then these are the free courses. So I've already pointed out that since February 2020, we have conducted more than 70 free courses in which more than 14,000 people have participated. And these are our services which we are providing in the form of the contract research, like virtual screening, molecular docking, MD simulations, predictive toxicology, and NGS data analysis, and genome editing. Now, these are the two other things which, if you might be interested in that, personalized training. Personalized training is something which we are doing for people who want training on specific topics. So you tell us, and we design the training program, and then we give you the training. Research project consultancy is for the PhD scholars. Like at some point of time, you know, PhD scholars, they're stuck, or even the master's student, they're stuck, and they do not know how to proceed. So they tell us, and then we design the future course of their project. So this is all about BDG Life Sciences. And as I have told you, I have taken only 20 minutes of yours. So you can take the screenshot. You can follow us on Facebook, Instagram, and YouTube, and LinkedIn. And then there is a dedicated team that works over there, and you can ask your queries, and then they can reply to your queries. So now coming to today's session, Envision with BDG Life Sciences. Today is the 8th session, and these are the free sessions which we are conducting under the banner of Envision with BDG initiative. And today's session is on brain-computer interfacing, and this will be taken by Dr. Haider. Now, just to introduce Dr. Haider is a lecturer in artificial intelligence for decision making at the School of Computer Science and Electronics Engineering, University of Essex, UK. Now, he has a PhD in computer science from University of Ulster, Derry, Londonderry, UK. During his PhD, he won the Best Literature Review Award sponsored by McGraw-Hill. He worked as a postdoctoral research assistant in brain-computer interface for both magnetoencephalography, that is MEG, and electroencephalography, that is EEG systems, at the University of Ulster, Northern Ireland. So I would not keep you waiting for long. So I'll just stop my screen sharing, and Dr. Haider, you are there. So now the session is up to you, and you can proceed from here.
Thank you, Asif. Thank you for the introduction, and welcome everyone. So I would like to share first of all my screen to you guys, and let's do it. So I hope you can see my screen, and today's topic of presentation, and you can say as a lecture, is about brain-computer interfacing. And Asif has already told you about me that I'm a lecturer in AI for decision making in the School of Computer Science and Electronics Engineering. And if you need to contact me, or you need to read my research, or you need to see what I have done, you can visit my webpage. It's sagihaider.com. And you can follow me on my Twitter and LinkedIn. If you want to contact me, feel free to email me on the email given on my website. And so the topic brain computer interfacing is quite interesting one, because I started my PhD with this topic. And I started my AI in year 2007 when I was doing my bachelor's. And by that time, I was very much interested into artificial intelligence and machine learning. So the journey started there. And today I'm still doing AI and machine learning and taking my research forward. And so the outline of today's talk will be, we'll talk about brain-computer interfacing, we'll talk about approaches to learn in brain-computer interfacing. So these are the methods which I have proposed. We'll go one by one over there, that what I have done. And at any stage, if you have any question, you can you can wait until the end of the talk, or if you want, you can unmute yourself and ask question. It's up to you. The session is open. So let's start. What is brain-computer interfacing? So brain-computer interfacing is basically a communication pathway, or you can say system that do not depend upon the peripheral nerves or the muscles. So I hope you guys know what is BCI. So BCI is what we do. We put a EG cap on someone's head, a participant head, and that head consists of several electrodes. Maybe it depend upon how many channels you have in the electrodes, or sorry, how many channels or electrodes you have in the cap. So it's totally depend how much information you want to capture from the brain. And then what we do, we capture those data, and we do some kind of processing on the data, and we decode the intention of the mind into computer commands. So as we move forward from in the talk, we'll see what is this, and it will become more clear to you. And this is a very nice definition by Wolpa that I have already told you, that brain computer is a communication system that do not depend on peripheral nerves and muscle. It's a communication between system and a computer. So let me give you a brief overview of this, that BCI system allow paralyzed patients, like a logged in patient. So like, for example, a person who won't able to communicate, maybe suffering from a stroke or full body stroke, any kind of stroke. And an example of this, like Stephen Hawkins, like means his body was logged in, he got a stroke, and then a BCI was designed and he was using to communicate. And then, so BCI system allow paralyzed lock-in patient to communicate words, letters, simple commands to a computer interface that recognize EEG signal and translate them into a control commands, which with the help of specialized algorithms. So I'm not a neuroscientist. I would say I'm not a full neuroscientist, but I'm a machine learning and AI scientist. So my job is to design these specialized algorithms. So what we do, we design these algorithms which can improve the performance of brain-computer interfacing, because whatever the system we design, we need that systems to be accurate, more like with a high performance. So my job is to do this part through designing the algorithm. Now, coming back to the next point, so non-invasive approach use basically there are different type of BCI. I will take you through that. So what we use brain electrical activities recorded at the scalp, and via EG. So that EEG which I have shown you in the last picture, that we put electrodes and record it. But now due to the because scientists want more clear data, noise-free data, so there is a new technology called MEG, which is instead of recording the electrical activity, we use magnetic like activity around the brain to record the intention of a person whom we are recording the data. So this is one of the work what we did. I will take you through to it. So this work I did when I was doing my PhD, and when I was doing my postdoc at University of Ulster, and we are in a collaboration with IIT Kanpur. So what we did, we have a team in IIT Kanpur, our collaborators, those who guys have designed this exoskeleton. You can see in this person hand, it's a kind of artificial hand, which helps a person to do stroke rehabilitation. So we help them to do a rehabilitation like exercise, because there is a theory behind it, and we want to test that hypothesis, whether this hypothesis is correct or not. So what we did, this triple IT team has designed this exoskeleton, and we were responsible for this brain computer interfacing. So we recruited quite a few patients in Kanpur, one of the hospital in Kanpur, and we took them and we placed the cap on their head and recorded the data and asked them to do several things and like several tasks of BCIs. And we did it for quite a long time. And like means what we were thinking that, for example, if somebody is having a exoskeleton, artificial limb on his hand, and what we are trying to do that if someone imagine that they are moving their hand. So for example, if a stroke person won't able to move their hand, they just imagine that they are doing that, right? So what they do, they imagine those things that, for example, at this stage I am moving my hand. So using BCI algorithms, what we do, we detect what they are thinking in their mind, right? So for example, they are thinking that they are moving their hand, but they won't able to do that. So once they imagine that they are doing a task, some kind of activity will happen in your brain, and we'll read that activity through EEG. And then as soon as we detect that intention from the human brain, what we do, we pass that command to the exoskeleton, and exoskeleton will move like this. So it will help you to do the exercise, because it is recommended in neuroscience that if someone is having a stroke, and the stroke is not too old, maybe it's a young stroke which happened like month or 2 months or 6 months ago, and if someone will do physical and mental practice together. So you are doing mental practice, like example, I'm just imagining I'm moving my hand, right? So I'm just imagining because I'm not doing it, right? But if a physiotherapist or maybe a helper can help you to, while you are imagining, to push your hand like this, right? So what will help, there is a theory that it will the neuroplasticity of the brain can increase and you can restore your moments. So what we did, we designed this system, because if somebody is doing their means having their exoskeleton and they are imagining, so we are helping to do mentally as well as physically also. So both things will happen together, and when both things will happen together, there is a higher chance of regaining the moment in the brain, means your physical moment, right? So which is known as neuroplasticity. So means once you gain your neuroplasticity, you will able to do the action because that part of the brain is dead. So this system we have designed, we have tested, and as we go later in the talk, I will show you that how we checked our performance and how this system helped, and we published a couple of paper on this research project, and it was a success. So now this is my team in University of Essex. So we are currently working under these two leaders. This is Professor Ricardo Poli, and this is Professor Renard Scherer. So these are our boss. You can say that our means world famous guys in BCI, and this is our BCI team. Here is Luca. He's another another guy with a great mind. He's Professor Francisco. He's also one of the leader in BCI. So we work together in to develop this BCI to make this BCI more advanced. So this is our BCI team. If you want to visit our page, you can see over it over here. So you can visit on this page and go and see our team and our work, what we are doing. Now, to take you through what is BCI, let me take you through a few points. A protocol means like what is the protocol of translating thoughts into action, because the ultimate objective of BCI is to convert your thoughts into an action. So whatever you imagine, can we convert those imagination into a computer commands or maybe a control commands? So how it works, I would like to take you through that. So as you know, the brain is full of neurons, and these neurons are connected to each other by axon and dendrites. So axon is a central part of that, and dendrites are those parts by which the neurons are connected. So your neuron connects with each other from and they form a kind of a super highway. So when the neurons, because the bunch of neurons are there, millions of neurons, and they once they connect together, what they form of a kind of a super highway. Super highway, why I'm saying that? Because they transmit information in such a speed that, for example, for example, I'm giving you when you touch a hot water or any glass of with a hot cup of tea, how suddenly you remove your hand? Because you touch something, your sensing neurons told you that this is not good, and suddenly you took the action. So can you imagine how fast the neuron communicates to each other, right? So this is why we say it's a very super highway for communication. And uh, and they communicate neuron to neuron, so one neuron will pass information to another, and they do it like that, right? So what we do, we use electrodes to detect these electrical signals which happens in the brain, and we send these signals to computer, because computer can then send these commands to external devices such as robotic arm, such as robot, or I will show you what other applications are. So then computer translate these electrical signals into data, which we used to control the computer or device linked to a computer. So this is a basic protocol of brain computer interfacing, right? Now, before moving forward, I would like to take you through the major historical event in BCI. So what are the major historical events on BCI? In 1924, Hans Berger, German neurologist, was the first to introduce human brain activity by the means of EEG. So they were the pioneers. They did that. And in 1970, research began in University California, Los Angeles. They began the research on BCI. And in 78, 8 years later, a prototype was implanted into man blinded in adulthood. And following the era of animal experiments, first neuroprosthesis device was implanted in a human brain in 1990. So first means surgery was done, right? And in 2005, Matthew Nagel was one of the first person to use BCI to restore the functionality lost due to paralysis. So in 2005, this BCI was implemented on a person or a stroke patient, and they, by doing the physical and mental exercise by using this BCI, what they did, they have restored the functionality of the moment uh, in the human who suffered with a paralysis, right? So it was a major breakthrough. And in 2019, Duke researchers successfully connected the two rats' brain together. So can you imagine that two brains of the rats are connected to each other, and they are transferring the information from one rat to another rat, and another this rat to, so vice versa, they are sending the information from one rat to another rat. So these are the major breakthrough events which happen in BCI. So now, what is the motivation? Why this BCI is designed? So BCI is designed to give disabled people a pathway to communicate or operate the prosthesis or controlling their wheelchair by their brain signals. And the target group, who are the people who are more means connected to this study or this research, the people who are suffering from ALS, and there are cervical spinal injury, stroke paralysis patients, cerebral palsy, and ampute. So these are the focused group, our targeted group on which this BCI was researched. And now let me show you what are the BCI applications. So this slide is very important because this is a kind of a block diagram which tells you how BCI works, right? So can you see a person over here, right? So in the person, this is a zoom part of his brain, and in brain there are several layers. So this is the skull is cup, and this is skull, and means there are several layers, right? So now the important point to note that is, you can implant electrode in the human brain. You can put it on the top as well, right? So if you put an electrode on the top, what we record, it's called EEG. That what EEG is. So we record EEG from there. But if we put in the middle, like not in the middle, but somewhere deeper in the brain, then we record ECOG, which looks like this somewhat. And then if we do a surgery and put deep inside the brain, then we got the raw signal, which is means we will read the neurons of the brain, right? So these are the process of signal acquisition. We'll talk later more about it, how they do and what type are they. uh, and then we, whatever the signals we get it from there, we convert them into a binary commands, which are like 0 1s, not binary commands basically. What we do record them in the form of digital signals, and then we do some kind of signal processing. So feature extraction, translation algorithm. So my job is this. So this is what I do mainly. This is my specialization. You can say that what I do, I design these algorithms, and these methods which take the brain signal and convert into the commands, right? And once we convert them, what you can control, you can communicate. So for example, by looking at the computer, you can write your message on the computer, which is quite handy nowadays, and which is very easy to do as well. And environmental control, you can attend phones, you can switch on the light, you can switch on your TV, you can do several tasks, right? Other application is moment control. In moment control, what we do, as I told you, in my study, we did neuro rehabilitation, we did rehabilitation. So you control a robotic arm, or you can control maybe a robotic leg. It's up to you, right? Locomotion, you can control the wheelchair, right? And you can control a robot as well, because for example, there is something lying on my bed and I want that robot to bring the thing to me. I got a stroke, so I can use my brain signals to tell the robot that can you go to that room and bring those things. So means it's several applications are there, right? And neuro rehabilitation, which is pretty important, because what we do, as you have seen in my work, that we did a neuro rehabilitation study and we tested our work on the patients, and we successfully completed our study. So which will help a person to restore their commands, you restore their moments, which is very important, right? So this is how whole BCI things work. It's look very easy over here, but it is not, because a lot of hard work is required, a lot of experience is also required, because at one go, if you someone says that I have EEG and I'm going to do this, no, it's not that easy. You need to understand different parts of BCI, because each has their own research area, right? So you have to carefully think what you are going to do. Now, basic steps in BCI. So basic steps in BCI is also one of the very important part, because I have told you before that as you see that a person head is there, you get the neural signal, and this part is known as signal acquisition, and we did digitize the signal, and we do feature extraction algorithms, we do classification. So because when you record the data from the brain, the data would be very noisy, because for example, once you are getting, or when you are recording your data, you imagine that you are doing your right hand, but back of the mind, you are thinking something else, right? So a mixture of information is there, and the people who meditate, those who do meditation, because they put their focus somewhere, so they are a good BCI user, because they know how to control their brain waves. So which is very important. So my part is, when we get the signal, how to get, or how to clean the signals, and how to learn machine learning algorithms based on the data, so we can better control these devices, right? So this is the part where I work, this is the part, right? And and we can control, I have told you different things. Now it's not only related to, you can say it's not only related to controlling devices, but nowadays people are playing games. Now this is a very big industry, playing games by mind, and there are several headsets, like VR kind of a devices, which comes where you can I mean, they are portable, you can just place on your head, you train that machine on your signal, and they are ready to use. You can play games with your mind without controlling a controller, or without any remote, or without anything, you're just playing game with your minds. And then once we do that, sometimes like means we need our user to also learn our feedback. Means, so what we do, because at one go it's not easy to tell the computer that do a task. So if there is something wrong, so what we do, we'll give a feedback to a user. So for example, there is a ball coming up from up, like a pinball kind of a game, and I'm trying to control with my mind. And what I'm doing, I'm trying to control, I'm thinking that the cursor should move to the right hand side, and I'm doing, and I'm doing, and suddenly means I was doing and it's gone wrong, right? So what we do in BCI, we send this feedback to the user on the screen, that you was doing right, and at the end, or at the very beginning, there was something wrong. so the human will also learn. So this is called co-adaptive BCI. So a human is also trying to learn that how to train a computer, and a machine learning algorithm is also learning a human behavior. So which is very important, right? So this is how this is means this portal this BCI steps in our whole framework of BCI. These are the steps which are pretty important to us. And there are different type of BCI, which I told you I will talk in later in the talk. So the type of BCI which we have, there are three different types: invasive, partial invasive, and non-invasive. So invasive is this when you put an electrode or any kind of a device by doing a surgery deep inside the brain, right? So when the surgery is required, it's invasive. And when you are not doing a big surgery, but you are just opening a little bit of skull and putting a scalp, and then you are just putting a simple kind of a chip or electrode not deep inside the brain, then you got ECOG signal from the brain. But when you are not doing anything to the user, you are just placing the cap, or you are just putting the patient into a machine and you are recording the signals. These are known as EG, on which I have worked. So I have experience of this. Currently I'm doing this because this is here we record electrical activities. And nowadays because of high quality of the data, I'm working on this MEG. I will talk later in the talk what is this MEG, so you can see. fMRI is also, but in fMRI you will not get that much of info, means you get detailed information, but the temporal resolution is not that good because you can't record the data for a long period of time. So but here you can play with it, you can control devices with these two devices, which is pretty important. Now layers and signal sources, as I have talked before. So you remember guys, we are doing EG. So when we are doing EG, it's non-invasive. So if it's here, this is non-invasive BCI. So if we are recording on the scalp, and if we are putting a electrode deep inside the brain, but we are not putting inside the neurons like that, right, then it is ECOG. And when we put micro electrodes like things inside the brain after the surgery, then we record this type of information, right? So these are the layer and the source of signals. So it's very important because all type of data is available in the web in open source. So you have to see where you can work. Here, the noise in the data is pretty big, or you can say the noise is large. So you have the signal processing algorithms play a major role, but this is easy to do. You don't need such type of ethical approval, although you need ethical approval to conduct studies on human participants, but you don't need to do a surgery if you are doing EG. But if you are doing these two things, you need to take a patient consent. Here also we are taking the patient or a person consent, but here someone needs to agree to go through a surgery or kind of thing, right? So these are the means challenges with these type of data, and this is the widely used, and you can find a lot of information available. So if I show you, I hope you guys can see my screen. So if I go through BNCI, so BNCI is a page which is called BNCI Horizon 2020. And here you can see some data sets which are free or available if you want to work on BCI and if you want to play with the data. These are the lot of different data available for free if you want to download the data and do BCI just to see how the EG looks like. So I'm just telling for your reference because you guys can download the data and do the job. Now, as I told you, non-invasive and invasive BCI. So in invasive BCI, we implant directly into the gray matter of the brain. So it means deep inside the brain. And in non-invasive BCI, we don't put anything inside the brain. We just put the cap and record the data. So this is how EG and MEG data acquisitions look like. So here you can see in what we do. So can you see electrode over here? This is the name of the electrode PZ, which is over this part of the brain, which is the back side, right? And what we are doing, we are having a reference electrode, because if you know electrical engineering or electronics engineering, you know what the meaning of this reference electrodes, because we are taking the reference with this electrode and recording the data. So the data would look like this. And Here you need to place the electrode on the human brain to record this data. But when it's MEG, you don't need to place an electrode on someone's head. Uh, Although the whole human head will go into a MEG machine, I will show you later in the slide that how the MEG data is recording. So here, instead of recording the electrical activity which we are recording over here, we are just recording the magnetic field which are being produced by the brain to see what thoughts are going in human mind. Now, coming back to EEG. So EEG is known as electroencephalogram. These are recorded by electrodes. What we record scalp recording of electrical activity of the cortex, which form a kind of a wave, as I've shown you. So this looks like this, right? So this is how EEG look like. And it's recorded in a microvolt, because you cannot imagine, we cannot expect the brain to produce means a recording in voltage. So it's a very minute thing. You need amplifiers to amplify the data. So whatever we record, it will come in microvolts, right? And we can record in multiple modes. We can record in bipolar mode or referential mode. It's up to you. What is your purpose? And this is the photo from one of our lab, and she's one of my colleague. And so we record EEG in our brain, in our lab, and it's not that difficult, I would say. But you need a device which is pretty expensive nowadays. Cheap devices are also coming, but they are not that accurate as a dense electrode system, because in the market there are quite a few systems like Emotive, or different companies are producing their devices, but they are not that accurate. The accurate devices are look like this, where you place the electrodes in a different part of the brain, and we record a good information, right? So MEG I was talking about, you remember I told you that I will tell you how MEG is recorded. So we record the magnetic activity from the brain. So in MEG machine is quite this big. The major disadvantage of using the MEG machine is that you cannot take this machine outside the laboratory. So one of the biggest question in BCI is that we are designing the BCI, but can we take BCI out of the lab? Because can we put a BCI system to someone's house, which we are doing currently? But this MEG system, which is a very accurate system, but the challenge in this that we cannot take the system out of the laboratory, right? But they are very accurate. So here what we do, we do squid, that is superconducting quantum interfaces devices are over here in this head part of the area, which directly record your magnetic field created by the electrical activity. So I hope you guys know, when several sources of the brain produce a kind of electrical activity, a magnetic force will be created in the brain, and that's what we record in MEG, right? And the data what we record, it's the format of the data is called femtotesla or picotesla. So Tesla is the unit of recording the magnetic field. And one of the other challenge of using this is uh, that it needs a shielded room to to record the data, which is very expensive to get, and this machine is itself is very expensive. So, and other issue I will tell you, so when I was recording the data, when we were doing the experiment, for example, there is a separate room, and in separate room recording is going on, and while the recording, we were sitting in the next room, and if someone will do like this or something, like you put something or you put your uh, feet on the ground like that, what will happen? You can see noise in the data. That's why these rooms are designed especially to avoid the noise, because this machine is very subject to noise. So we have to be very careful while recording the data. So a comparison in EEG and MEG, and this is very good picture which I found on the web. The two things which we actually want from the data need to be good in spatial resolution and in a temporal resolution. So you can see the MEG data is okay and good also in spatial resolution, but EEG is good, right? But these two what we talk about, but their temporal resolution is not that good, right? And this fMRI PET, they have a good temporal resolution, but their spatial resolution is not that good. But here you can get a time series data, but in these type of signals, you don't get time series data, you get an image from the brain, right? So these two are basically a time series information, but these are not a time series information, right? Because their information is not that great, right? Means, but they are used for different purposes. Now, EG and MEG comparison. So signal magnitude of EG is 10 microvolt, and here it's 10 Tesla, F Tesla, right? Measurements are secondary currents. Here are primary currents. Signal purity is distortion by skull and scalp, and little effect by skull and scalp. So it's not that effective. So it means it is effective, but the scalp and skull is not playing a major role when you are recording MEG, because it's penetrate and uh, the signals come out because it's being generated outside your brain, right? So temporal resolution is 1 microseconds, and oh, sorry, 1 millisecond. Here it's again 1 millisecond. Spatial resolution, you can see in MEG the spatial resolution is quite less than EG. Both experiment moves with subject. This is the one of the major difficulty, that MEG cannot go outside the laboratory. So this is one of the major disadvantage. Dipole orientation, it's tangible and radial. Here it's tangential better. So it is just for your purpose, that what are the difference between two. In some cases this is good, in some cases this is good. It's depend upon your study, what you want to do. Now, BCI paradigms. So this is very important to you, because you need to understand what you want to do. So there are different BCI paradigms. The first paradigm is P300. I hope you guys can see what we are doing. So I will go through in detail. This is just an overview. So we are using a human eye, and we are recording the brain signals to control a keyboard, or you can say to write something on the computer. Here again, same thing, but with a different approach, which is known as SSVP. I will come back to this later. And SMR, that is known as sensory motor rhythm. So which are being generated when you do something like such as move your hand, move your tongue, or move your feet, or move your body. So some activity generated in the brain. I'll come back to this later, because I'll go through each one, because you understand means what is this BCI paradigm. So let's go to the first paradigm. First paradigm BCI is P300. So P300, as the name suggests, why it's P300. So P300 is a paradigm where the user is requested to focus on something. So for example, you have a screen in front of you, and like a specific letter on the screen. So when you go to eye doctor, you see there are some alphabet written in a random order, right? But random order, but their size are different. Here the sizes are not going to reduce, the sizes are going to be means correctly fine, right? So when a letter is suddenly highlighted, so what will happen, I'll show you in the experiment. I have a video for it, which I found yesterday. So what will happen, so on the letters there are lights moving like this, and when you focus on a specific letter, for example, on this monitor there are 26 alphabet. On among those 26 alphabet, I want to focus on H, and I'm looking constantly as H for maybe 3 to 4 seconds. And when quickly the light pass through H, there kind of some activity will happen in my brain, and in EG you can see that activity. And when I was focusing on that H, the activity in the brain will come after 300 milliseconds, right? So that's why it's known as P300. So by using that, you can see over here that event related potential is being generated in the brain, and there is a sudden drop in the voltage just after doing a task while I was looking at H. Once the light will pass from the H, just after 300 milliseconds, there will be a drop in my voltage in my brain, right? So the computer will detect and see that this person is reading this letter, right? So let's if possible, let me go through this video. Maybe if I can, no, I don't want to leave it. So how, if I copy this, and maybe, I hope you guys can see. So this is what, there is no sound in this video, so This is how the EG cap and their amplifier. So this is the EG cap, this is amplifier, and this is a connector where you connect your EG. So the signals will come from here. This will go to this part, right? And then amplifier will amplify those signals and you record those signals, right? So now see, this is the EG cap setup. This video I found somewhere on the internet. So once someone is placing a cap, you have to put a gel inside it, because once you put the electrode and the gel will reduce the conductance, means like because you need to read the brain, right? So you can see that these are the signals which are coming from the brain, and this type of light is going through, and this person is going to write vision. So he's currently focusing on V, right? So you see real time is P300 spelling. So this is what he's trying to write. So he is focusing on V, and so see, now the one trial is over. Now he was focusing on I, and as soon as the computer detect he's focusing on I will be written over there, right? Now S, he's focusing on S. Yes, so similarly he will write the whole thing. I don't want to wait. So you see, by the end he write vision. So this is how P300 BCI work. uh, so this is what a P300 BCI paradigm is. So if a person, so for example, lying on the bed and have a stroke, can't do a task, and they want to do send a text to someone, or they want to write a message for you, which because they can't able to speak, this P300 BCI is pretty good for them, right? The next type of BCI is called SSVP, which is called steady state visual evoked potential. And it is an example of paradigm where element will flicker steadily on the screen, but with different frequency, right? So what will happen over here, that you have different portion of your screen, and each portion of the screen is means each letter on the screen is flickering. Flickering means it's coming and going with a different frequency. So for example, one specific block is moving, or you can say flickering, with a frequency of 10 hertz, other is doing at 20 hertz, or other one is 30 hertz. So when the user will focus on a specific element, the corresponding frequency means frequency will become stronger in your brain, right? So for example, if I'm looking at one block which says open, and which is flickering at maybe a 10 hertz, and when I was looking at my means with my eyes over there, the 10 hertz will become active in my brain, right? And then by using that you can select multiple options. So for example, when you go to a mobile phone or something, for example, you open an app where a few options are there. So first option is this, second option is this, third option is this, fourth option. So you want to go through the third option. Third option is there, you select it, right? And then another four options open, so another four options open. So this type of study if you want to do, and then this type of thing is called SSVP BCI. I will show you this as well with the help of a video. So let me show you. So see, so first I will tell you what is happening. The person is trying to control this robotic arm, right? And you can see 1 2 3 4 something is going to be written over here. So wait for a while. So see, so he's currently looking at one, so only one finger is open. He is looking at two, the second frequency is open, sorry, second finger is open, third finger is open now, and then fourth finger is open now. Now he's again closing them back. Two, so now two will be closed, and now one will be closed, right? So this is SSVP based BCI. So if someone is having a stroke and they want to communicate, this is another type of BCI. And this BCI, which is called motor imagery, and my whole PhD and my whole work is on this type of BCI, which is called motor imagery. So motor imagery is based on the user kinetically. What does it mean? So for example, I just found this picture which is very useful. So can you see a lady who's trying to ski, and while skiing, because she was skiing, so she's activating this part of the brain. You can see I'm just highlighting it. This part of the brain is getting active, right? Yeah. But when someone is having a paralyzed, or maybe they have a stroke, but she's not skiing, but she's just imagining that she's skiing, right? So here the thoughts from the brain is forming that, for example, uh, yeah, so means she's just imagining that she's skiing. So not that dense activity will form, but some form of electric activity will form in the brain, right? So for example, if I'm having an EG cup on my head and I want to move my right hand or left hand, whatever, so if I'm doing my, I'm won't able to move my left hand, and I'm just imagining that I'm moving my left hand, what will happen when I imagine, when I move my means left hand, the right part of the brain in the motor cortex area get activated, right? So but when I won't able to do that, and I just imagine, I focus that I'm doing, then also a form of activity will be formed in the brain. So this is known as motor imagery, which is related to the motor cortex. And what will happen over here, that you can help someone to do rehabilitation, like controlling the robotic arm, or maybe you can drive a robot. So for example, your robot have just 4 options to go forward, left, right, backward, and you just imagine that, maybe I'm just imagining that I'm moving my left hand, and I told my computer that when I imagine left hand, my robot should go forward, right? And when I imagine my right hand, my robot will go to the right hand side, right? And when I imagine that I'm moving my feet, my robot will go to the left hand side. And when I imagine I'm doing something with my tongue, like moving my tongue, but I won't able to move my tongue, my robot should go back, right? So when this type of BCI is done, this is done through the help of motor imagery. And we have another video, I believe, for this. Let me stop this. Yep. So, oops. Yeah. So, not this one. Selection is, sorry. 1 second. Yes. So, maybe let me. So, this is how motor imagery. So this guy is trying to play a pinball. He's not moving his hand. He's controlling the pinball through this. So he's controlling the falling ball towards left and right. So now the right one is flickering. So he's imagining to moving his right hand, and ball is coming to the right. So he's trying to play the computer game through the motor imagery. So you can see he's just imagining, he's not doing anything. He's just controlling this smiley, or you can say a ball towards left and right. And this is what we do with the motor imagery based BCI. So currently he's doing it right, which is going, and this is how it's done, right? So, okie dokie, now move forward. So yeah, so let's dive a little bit more into that, what happens in the brain when we do something, right? So the patterns in motor imagery can happen. So imagination of the moments, what we you need to detect. So for example, we have 2 tasks, left hand moment and the right hand moment. So what will happen in the brain, that for example, when I imagine or I move my hand. So for example, if I'm moving my left hand, the right part of the brain, it's got activated. And the electrode, so you can see here maybe C3, CZ, and C4. So each electrode has a name, right? A number are tagged with it. So C3 is on the left hand side and C4 is on the right hand side. So here is C3. So this is your C3 electrode, and this is your C4 electrode, and C3 plot is this and C4 plot is this, right? So when I imagine something on my right hand side, right hand, something will happen in C3. And you can see when something is will happen in C3, right? So the means the plots, the electrical signals which we are getting, you can easily distinguish between one electrode activity and other electrode activity. So here, if you are doing something from your left hand side, your C4 electrode voltage is dropping. So what machine learning classifier will do, we will try to learn this gap between these two lines, or you can say the curves. So this is what we are trying to learn with the help of machine learning algorithm, that if someone is doing something, how we can distinguish the gap between these two, right? So this is how this BCI is done, right? So now if I zoom it little bit, you can see uh, that these are the activity in the brain. So if you plot the topo maps, right, you can see if someone is doing a movement of the left hand, the right hand part of the brain is getting more dense. So this is the right hand, right part of the brain when you are doing left, and when you are doing right, a left part of the brain is getting activated, right? So this is how a topo plot sees, and topo plots are the kind of a plot which we do for brain, and this is how we read the electrical signals. But behind this there are a lot of signal processing. It is not that easy. You need to go through different signal processing methods. First you have to filter the data into a specific frequency band, and then you have to apply feature extraction method, which help you to which by the help of that feature extraction algorithm, you can separate the data between left hand and right hand task, right? So this is how it's look like, means just for your overview. And this is how the head of means electrode cap systems look like. We called it 1020 electrode system, where 128 electrodes can be placed on the human brain. And you can say in MEG based BCI, you have, instead of having 128, sorry, I cannot write with this. Here is 128, and here we have 306 channels there. So which includes, I think, 206 gradiometers and 100, no, yeah, it's having 204 gradiometers and 102 magnetometers. So if you zoom it, you can see that means different squids are there which were placed in the brain, not in the brain, outside the brain, in the MEG system. So this is how we do motor imagery in the BCI, in this BCI, right? Now let's come to the part where I researched. That was just a background. So challenges with EEG and MEG signals are there, that they are very prone to noise and outlier. So you can see, what does it mean by noise and outlier? So when the electrical recordings were going on, or maybe when someone is being recorded for their brain signals, uh, for example, when you are doing a recording and someone blinking their eyes very frequently like this, like you're flickering your eyes, or maybe someone passing around you and they tap their feet, or means whatever. So you can see this particular part, you can see they become noisy, right? So we want to get rid of it. So to getting rid of it, you need specialized algorithms, or you need a specialized signal processing method, because if this noise will be there in the data, you won't able to decode the data. So you won't able to decode the EEG signals. So to decode them, you need a good signal processing methods, which will help you to clean your data, because machine learning algorithms, they are not humans, they are not intelligent, but we make them intelligent, right? So our job is to give them a clean data, because if we provide them a clean data, they will do a good job. But if we'll not provide them a clean data, it will not do a good job, right? Second problem is high dimensionality. What does it mean by high dimensionality? So high dimensionality is that we have the data which is coming from brain, and for example, you are recording from 128 channels, or maybe not 128 channels, say you are recording through 50 electrodes, right? And if you're recording through 50 electrodes, mm, you need a handsome amount of information, because when 50 electrodes information is coming, they are coming in a 50 variable, right? One variable, two variable, three, four, five different variables are coming, right? And once you give that data to machine learning, uh, we don't want much information in the column, rather we want much information in the rows, right? Because if the number of rows are bigger, sorry, if the number of columns are bigger than the number of rows in some ratio, the machine learning algorithms won't able to work nicely, right? So, so we want to get rid of this high dimensionality. And if you are a little bit familiar with the data analysis and machine learning or a little bit of artificial intelligence, there are few methods which were used, which we also used for removing the dimensionality of the data, which is known as PCA or ICA. Different methods are there, common special patterns, different algorithms are there to get rid of this. Another issue is small training sets. BCI systems are not designed ready to go, because if you get a BCI system, you need to first record the data and to train the machine, which is known as calibration stage. So first you record your data and give it to your machine that this is my data. First learn that how my data looks like, right? So for example, we at least we need half an hour of recording to get a good machine learning classifier which can decode our thoughts. So the problem is that every time you come, you need to train your machine, because human brain is a dynamic machine. So for example, I wake up in the morning and I have a BCI system. I record my data, I trained a classifier and it was working well. And for example, at that time, the accuracy was 80%, right? And later in the afternoon, what happened that I got a call from somewhere, or I got tired, or I was watching a football match and my favorite team lost, whatever it is, the reason I feel low, my mood is not good. What will happen? The same person, if you record the data again, the properties of the data will change, right? These are the dynamics of the brain, because the data will change, right? So what will happen when you record the data in the morning and the evening, it's the same person data, they don't look same, right? So what will happen when you train a machine learning algorithm? You learn a particular classifier, or you can say a hyperplane, and in the evening when you come to test the system or to use the system, that system won't able to work because of this, right? So you want to get rid of this issue also, which is very important. And this is what my PhD is upon. So I have to get rid of this issue, not completely, but to increase the performance. So I will slightly zoom my slides so you can easily see. You can see two ellipses on the left hand side. So this is ellipse 1, this is ellipse 2. And ellipse 1 is the imagination of my hands of left hand, and blue right hand side ellipse is the features which I have extracted from my right hand, right? And it was the morning session. And what I have learned, a machine learning classifier is a black hyperplane, because using this black hyperplane, I can easily divide these two ellipses into whatever that blue dots or things are coming over here, it's become left. And whatever the data is coming here, it's become right, right? Now, when I came in the afternoon, what will happen? I recorded my data again, and you see the ellipses has gone totally wrong because of my mind wandering, or maybe I'm tired, or maybe the electrode cap which you have not placed in the correct direction, or maybe they are slightly misplaced, or the electrical gel the conducting gel which you have placed, that is not right. So there are several challenges, right? So my job was, how is it possible to reduce to reduce the gap between this blue and red ellipses, right? So, and this shift in the data, so for example, this is the morning session, this is the afternoon session, the shift in the data is known as covariate shift. So covariate means input, and shift means that input has shifted, right? So when your input has shifted, so for example, a best example is this. For example, I'm teaching you geography, right? For example, if I'm your teacher, and whole semester or whole term I was teaching you geography, and when you came to an examination, I gave you a mathematics paper and I'm asking you to solve this paper, would you able to do it? No. Same as machine learning classifier, my morning data is looking different, right? And when it's come to the evening, I'm giving you something different to classify. How would you able to do that? So same thing happened with a machine learning classifier also, that they look different, right? And same, the problem is same. This is EEG data, and I see the same problem in MEG data. So this is my morning data. This is left, this is right, right? And I trained a classifier this. When I came in the evening, this is my left, this is my right. So what I have learned in the morning is this, and now in the evening, I need a classifier hyperplane which goes like this, which is not possible. So means any machine learning algorithm you can bring won't able to do a good performance, right? So this is what my PhD is, and we propose several methods to reduce this. I'll talk it about later when we'll come in the talk. And so these are the issues. So challenges, the classical learning system, I told you, they are built upon that the input data distribution of the training stage and the testing stage remains the same, but it's not, because whenever a new time you come, your brain signal changes. And this problem is also known as non-stationary problem, because we have a time series data which is coming from the brain, and that data is non-stationary because it changes over the time. So the learning become difficult, and the system performance will degrade over time. So because in the morning I was this, in the evening I was this, so we have to get rid of this, right? So now what I worked on the method whose big name is called non-stationary learning. And in this non-stationary learning, we found this map. This is whole machine learning, guys. Don't, if you are a biologist or something, please don't feel bad, because I just want to tell you that how it's connected. Uh, so the problem is this, there are several domains where you can work on. This is all AI and machine learning. So what I did in my PhD, I touched these two areas, and I implemented these two type of AI and machine learning in my BCI to make it better, right? And currently I'm working on this part of the area, right? So this is a big mind map of learning in non-stationary environment. This is only not limited to BCI, it's applicable to everywhere. So for example, wherever the data is coming in real time, maybe it's genome data, it's DNA, not DNA, maybe uh, any weather data, any finance data, or sports, whatever data which you are getting in real time, subject to non-stationary, right? And if it's coming in real time, it's going to shift, it's going to change. Then how do you are going to tackle it? So this is what I did. I worked on these two approaches, which I'm going to talk you a little bit. It's not AI in machine learning. They are just, I'm just going to tell you what I did. It's not that technical, but I'll try to explain with the best of my abilities. So, and you are free to ask questions, right? So now, route to non-stationary, I told you I have to design few things. So I can show you this figure. So this is what the issue is. So here, human participants are there. We are getting the raw data. We are extracting the feature. We are doing dimensionality reduction. We are filtering the data. We are feature extracting the data. We are doing data aggregation. So what I proposed to put or introduce a new block in the system, which is known as covariate shift detection test, and then implement a classifier. So the whole scenario is this. Let me explain you in a layman term, because otherwise it's going to be a little bit technical. Uh, so for example, there are two approaches to adapt, because if you don't adapt, you won't able to perform better. So adaptability in BCI is very important. Like if anything changes, you need to adapt it. So, and if you need to adapt, you can survive, right? So but there are two ways of adapting. One way is known as passive adapting, or you can say passive approach, and other one is called active approach. So now coming back to the late term point, consider this scenario that there are two type of people basically. Those who want to stay active, they run, they do exercise, they take multivitamins, they take means they take care of their health. So they are passive, they don't want to get sick. So every day do they exercise, they take pills, they take multivitamins, they take care of their body, right? So these are passive people. But there are people who are active in terms of not active in the physically, but their approach is active. And what is that active approach? That, for example, when a person gets sick, then only they will get to the doctor, right? So then only they take medicine in their body, or they take care of their body, right? So same approach I applied it here. I designed two BCI, one which adapt to signals over time. So for example, every time a person come, I take their data, right, and train a BCI. Now, other type of BCI which we have designed is active BCI, active approach based BCI, where what we did, uh, by introducing this layer, I was passing the feature extracted data. So the data which we got from here, we are extracting the feature and then we are passing it here. So here I have designed a statistical algorithm which detect the change in the data. So changes, what does it mean? The statistical distribution of the data will change over time, right? So for example, if your data is normally distributed, normally is defined by mean, standard deviation, and you can take other higher order statistics. But there are some statistical method to detect a change. Change detection itself a theory of a science, where it's a field of a research. So I have gone through several papers and I designed an approach for it. And yeah, so what we did, we implemented this method over here. So wherever, for example, I came in the morning, I recorded my data and I train a BCI, and in the evening I'm going to use it. So now when the evening I will come, this shift detection test, as soon as I started recording the data, if this change detection test will tell the BCI system that, hey, Heather's data is not similar as in the morning, it's a time to ask Heather to give a new data or adapt kind of a methodology or a change your algorithm, which can minimize the gap in the data, right? So this was the approach what we did, right? And for that I have to design a few things. I have to design first this method, that how to detect a change and then how to adapt. So first I have designed how to detect and then how to adapt. So to detect the change, I have designed this algorithm which was published in Pattern Recognition Elsevier. uh, and it was pretty mathematical approach. It took me nearly 12 months to design this algorithm and test and publish, not publish, just to test and peruse the results. And by the name it says EWMA. So EWMA is a well-known method in mathematics. And what was my contribution? My contribution was that I I I designed this type of a figure, and then I proved a kind of a lemma which tells me that if your data is coming from a time series signal, and if your data is autocorrelated, what you can do, you can predict a one step ahead error, right? You can monitor the error, so you can predict the one step ahead uh, step of that particular data. So what will happening when we are predicting the one step ahead of the data, we are matching it with the current data and we are plotting the error. So what will happen when we plot the error will form a kind of a Gaussian distribution. And whenever the Gaussian distribution goes out of the control limit, we'll tell that there is some change in the data which is happening. And that time the BCI will alert the system that now something is changing. So let's either retrain or take a new data or use a new approach to get rid of this problem, right? And there was other thing also, that there was no time delay, but that was very good. There was no assumption on data distribution. But the problem is this: when I have designed this test, that a high rate of false positive was coming. And so to overcome this challenge, what we did, we introduced another layer inside the test. But the issue that came from the last time, that we have reduced the false positive, but the advantage over the previous approach was this: in the previous approach there was no delay in the detection of test, but here we are getting a delay little bit, maybe a sample of a delay, maybe like 10 points or 20 points when you gather the sample. So a little bit of delay is there, right? And these are some of the results which we have published. So we have tested out it on toy data first, and then, sorry, we took the data from the real time, not real time EEG data, which are publicly available, and we tested our approach, we published, and it was well fine. So the first part of my PhD was this. Second part was to design a co-adaptive BCI, co-adaptive like covariate shift. So the approach was this, that I have to design an algorithm once the change is detected, how to adapt, right? So what we did, I have designed an adaptive ensemble algorithm. Ensemble is a method uh, where not a single classifier, or you can say a predictive model is being learned. We learn multiple models. We learn different models. So instead of having a one judge uh, to judge the input, we are having multiple judges, right? And we are taking the input, we are providing the input to multiple judges, and multiple judges will combine their judgments, and then we will decide, right? But it seems easy, but because there was something which I have designed also, I have introduced a transductive approach also over there. uh, so my whole approach looks like this. So when we get the EEG data, we band fast filter the data, we do the spatial filtering. So here what I have introduced, my test. So the test which I have designed uh, is coming over here. And once we get this, we train a classifier, and when the testing phase is there, we do same thing, but the test will tell whether the data is changing or not. And if that data is changing, we are going to adapt the change in transductive mode. So it's pretty machine learning, it's over there, but believe me, it's worked, and that work was published in neurocomputing. And if you want, you can go to my Google Scholar and read about this paper. And the performance was good, and you can see clearly that in previous approaches the performance was 73%, 75, 77, 75, 71. And in my approach, what we have proposed, in my passive approach, the accuracy dropped in first scenario, but in second scenario we have improved by 4%, and in third scenario we have improved by 5%. And similarly over here in the last approach, we have nearly improved by 5 to 6%, right? So the approach worked, it was working well. And the best part is this, it doesn't work on the offline data. We have also implemented this in real time BCI, and I have tested it by myself, and it was working well, right? So this is what we did. So how much time we have left? I think 1 hour, Asif. Okay, I think he's not there. Anyway, so CSA based CSA like covariate shift adaptation in neuro rehabilitation. So this is the study which we did in IIT Kanpur. So we have designed a co-adaptive BCI, covariate shift adaptation based BCI, where this hand exoskeleton was designed by IIT Kanpur. So whatever I proposed in my PhD, we have implemented in this study. We have implemented everything in online. And what we did, the system overview, I will tell you what we have. We have used GRAS. There is a company called GTech, which is a pioneer in BCI. They are based in Austria, sorry. So we have used their system, and we have used MATLAB simulating environment to create our whole means study. The exoskeleton was developed by IIT Kanpur, and it was developed using nylon-based lightweighted material with a rapid prototyping. And you can see it has grip also, so you can. It's a three-finger exoskeleton, so you can place your three fingers. We have caps also inside. You can see you can place your finger over here, right? And there are some force sensor also, which help you to do motion. So for example, if the sensor will see that you are trying to put some force, it will detect your force and help you to do the exercise also, because we have several hypotheses to test. So we did it like that way. And you can see the paradigm is look like this. So the paradigm is look like this. So when we train the machine, we have given a kind of a thing in someone's hand, and we are reading when they are doing a movement. So we are trying to ask some patient to open and close their hand, which look like this. So you can see at 4 seconds somebody is holding her hand, and then partially open, 5.5 second half open, 6.5 second is fully open. So we have a 8 second of a trial where we give first 3 to 4 second a patient to relax and to get ready. So you can see on the screen a message will come as a get ready, and 3 seconds the instruction will come that you have to control the, you have to hold the remote, and then, right, then they do it for several seconds, and then they do it like this, right? So we asked means our patients to do this type of exercise, not, yeah, so they are doing like this, so we are recording their data, and then we trained our classifier. Later, when the patients came, we put exoskeleton on their hand, and we are just reading their intentions. So if uh, we detect that they are trying to close their hand or open their hand or trying to do the exercise, the exoskeleton will help them because they won't able to move their hand. So exoskeleton will help them to open and close their hand, right? So this was done. And what was the protocol and data acquisition? So we have 2 runs of 40 trials. So before our patients come and we test them, we'll give them 2 runs where 40 times they have to do like this, right? Imagining, right? So they are just imagine because we want to read their data to train our machine learning model. Once the model is trained, uh, we do the job, right? And we used 12 different electrodes from the human brain. Instead of using having 128, we just used these electrodes which are near your motor cortex, because motor cortex is responsible for reading the motor activity near your motor cortex. And we have also placed EMG is electro based, which is placed on your hand, because we want to read your muscle signals also, that how much effort is doing on, right? So muscle moment we also want to read. So this is what we have done. We have sampled the data with 512 frequency hertz. We have filtered the data from 1 hertz to 100 hertz, and then we put a notch filter at 50 hertz because of the electrical activity which we have in the Indian circuit. We have to put a noise. We have to filter the data over there. And we have 10 healthy volunteers. We have 10 patients also, and we recorded their data. We have seen that none of the person have epilepsy history, right? We did some test also, their mental task test, that whether they are good or what is their score, right? So we did it like that. And we have a written consent from them. And this is what the score was there. So you can see that when there was no adaptive BCI, when we were using a simple BCI, their score was 75%. And when we implemented our system, their accuracy is increased by 6%, which is 81%. This is with a healthy group. And if you go to the patient group, uh, so the patient group is having the accuracy with a non-adaptive BCI is nearly 70%. And when we have used our system, it is 75%, which is pretty good. We have improved the accuracy by 5%, which is a good improvement. And yeah, so this was one of the study which we did. And we have another hypothesis that during the rehabilitation process, uh, a physical practice by a mental practice, when we combine both, right, uh, the chances of regaining the neuroplasticity is increasing. Means there should be an improvement in that. So we did that study also. And we did, means these all are the protocols which we have followed. Means everything is there. If you want to go and read this about this work, you feel free to go to this paper. There are everything is mentioned over there. And I just want to share the results with you. So you can see over the month, we started when they have 60% of accuracy. And when we finished our study in session 15, so that means after nearly 3 months, they have improved their accuracy from 65% to nearly 80%. So these are the patients. So patient 4 is nearly 80%. And there is a great improvement in patient 1. So you can see he's nearly 93% accurate now to control the robotic device, right? These are their topo maps, and these are their demographic information if you want to have look. And these are their art scores and different measures of their success. So grip strength, we have art score. So if you want to have a look, you can read the paper. And last thing, not the last thing, second last thing, I was not only limited to simple machine learning. Recently, I started doing deep learning for CSA, and in deep learning, because you know that deep learning or neural networks is a black box where you don't know what's being learned or something. So what I have proposed recently, I have proposed a method which is called unsupervised domain adaptation. Here, we are going to adapt the data without knowing the labels, and we did a great job here also. Previously, we were having just the accuracy of 78%, as you can see over here. But using our approach, we have increased to 83%. And this work was published in IJCNN, which is a very good conference, and in AI and machine learning. And last thing which I want to tell you that I have talked about EG, and now MEG data set we have, because MEG is rarely available. I think in India only 1 or 2 machines are there, and in UK we have nearly 16 or 17 machines in universities or hospitals, and we have recorded their data which look like this. So, and we are releasing this very soon. My work is under consideration, so we have submitted this work in Nature Scientific Data. And if you want to start your PhD, or if you want to work on EG or MEG, I would say this is a high time, because we are releasing this data. Nothing has been done on this data before, before releasing. So if you want to start your PhD, I would say consider this data. Nobody can stop you to publish. I'm 100% sure. This is a very good data, and we are going to release this data in open source. And if you are interested, feel free to email me. I can give you the codes also, just to do with the basic analysis, and feel free to play with this data, right? So thank you. This is in the summary that I took you through EG and MEG, what I did, how I managed non-stationarity in the data, what is BCI, and what we did in neuro rehabilitation. Yeah. So in the acknowledgement, I would like to thank my funders and my funding bodies. These are the list of my collaborators. These are the funding bodies. And yeah, thank you. I'm open to the question.
Thank you, Dr. Haider. It was very nice speaking to you, and you have really explained this very technical topic in the simplest way. So thank you very much for that, and it was a pleasure to have you. And I think I have got only one question, and the question is like. How NeuroPerfect plus EMG 2000, and then these are the name of the devices, Sleep Care and Machine 32. So what the question is that these machines like a channel polygraph machine, how they work, and can anyone misuse for their own benefit?
Misuse? What type of misuse? Means like, see, I told you there are so many suppliers, right? There are so many suppliers. Each machine is good on their own. It's up to, because you cannot say that, for example, when mobile came, there was Nokia, right? Now there are so many in the market, right? Or Motorola, right? So each have their own good things, because when you buy the device, right? So for example, the first thing you have to see the maintenance, right? So for example, we are using GTech. I use GTech because the maintenance is available within the UK. So please check before buying, or think that if something will happen to your machine, you don't have to ship to Europe or maybe US or somewhere, right? So that is the most important thing. And if you want to do the study, see, first search or do your survey that which type of literature is available. For example, for motor imagery or these type of thing, I use GTech. And because most of the studies which are available online, their tools, their open sources, everything is available, right? So means before buying anything, I would say see what codes are available, because there are a lot of coding involved, right? And a lot of experience in signal processing, and working with patients or working with healthy participants is required, because you don't want to harm them. And in terms of misuse, like misuse is there, means there are several type of misuse possible, right? Where you can, but that misuse means, but you cannot misuse it. I'm sure it is risky, right? You cannot leave a patient with a robot. But
like more comment has come up. It is saying like ENG for neuron damage.
Neuron damage, you know why it will damage? Why it will damage? It's reading, it's not transferring anything to your brain. It's just a sensing device which is sensing something from your brain. It's not transmitting anything which will go inside your brain.
Yep.
Exactly.
So I think that is all. And just let me check out if we have any questions on our YouTube channel. No, we just have a comment that please explain by zooming it. Okay, so then that is a very Haider, your yeah. Okay, fine. So we've sent you the feedback link, and you guys can send us the feedback. And the link is, as you all know, that link is valid only till 1 p.m. tomorrow India time. So before that, you need to fill that feedback form and provide us your valuable comments, and we will email you the certificate. So once again, thank you, Dr. Haider, for your precious time. And it was a very nice session, and we wish you good luck for your future research. So thank you. And I thank you from all the participants who have joined, and obviously from the staff of the BDG Life Sciences, and personally I do thank you as well. So thank you very much, and see you soon.
Thank you.
Thank you.